She's ready now. Had to loosen up the moment. Well, welcome everybody. Today's first of a PSRC doubleheader. Jessica Jang is going to talk about her fascinating study on the big skate, Beringraja binoculata. And for those of you that don't know, today's Flat Shark Friday. It's really appropriate. A flat shark today. So, Jessica?
All right, thanks, Dave. So let's start. So the layout of the presentation, I'm going to start with introductions, objectives, hypothesis, methods, results, and then discussion, and then wrapping it out with a conclusion. So if any part of the presentation, should you get lost or fall asleep, there will be icons in the top right-hand corner indicating where we are in the presentation. So skates are really diverse. They're one of the most diverse species of elasmobranchs. This includes sharks, skates, and rays. There's over 300 confirmed species, with many still waiting to be discovered. So they are dorsally ventrally flattened, which makes them susceptible to benthic bottoms, such as fishing practices like trawling and longlining. Because of their similar morphology, it makes it really difficult, as most fisheries management group them up in a generic skate category. However, recent studies have shown that species can have different sexual maturity ages and sizes and even breeding season. So for the species on top, this is the little skate, Beringraja inrincia, which is known to have sexual maturity between 7 to 9 years. And then you have the species on the bottom, the rough-tailed skate, which is Beringraja tricura, which can take up to 21 to 24 years to sexually mature. So these variabilities stress the need to understand how each species' life history differ in order to properly manage them. So a little bit about, well, no, it doesn't play. Okay, a little bit about the skate reproduction. So skates are oviparous. They lay a keratin-like egg case, which the embryos rely solely on the yolk. These egg cases can be laid almost anywhere on the sea floor, from sandy bottoms to rocky reliefs. Incubation time for some species can range from 3 months to even 3 or 4 years. So this was supposed to be a video of showing a nursery site of a, nope. Oh. Okay. Doesn't work? Okay. So this is actually a clip of the Zamchung Canyon, which is off the coast of the Gulf of Alaska, off the continental shelf, and it is a nursery site to numerous species of Beringraja. And you can see that all these little capsules are of egg cases of several different species. So in 2012, a new genus was introduced in the family of skates. So this is the Beringraja genus. 2 species were in this genus, Beringraja binoculata and Beringraja pulchra, which are the big skate and the mottled skate. These characteristics included similar clasper morphology, which is the male's external sexual organs, egg case morphology, as indicated here, and the ability to produce multiple embryos within an egg case. So only these 2 species have this ability, ranging from 1 to 9 embryos per egg case in Beringraja pulchra. However, in 2016, Last and al. Added 4 more species to the genus, which don't exhibit the multiple embryo trait. So a little bit about my study species, the big skate, Beringraja binoculata, which I will refer to now as the big skate for simplistic reasons. So they're found from the eastern Bering Sea to Baja California, and is considered the largest species of skate that we have here in the Northeast Pacific coast, reaching up to 2 meters in total length. And so total length refers to the tip of the snout to the end of the tail. So this is just showing you how big they can get. They are known to have up to 8 embryos per egg case. And though the IUCN, the International Union for Conservation of Nature, the IUCN, has assessed the species as least concerned, its sister species, the model skate, is now considered vulnerable with at least 90% of the population overfished. So there is no direct big skate fishery for this species here in California, but it's commonly sold to Asian countries for human consumption. So this poses a problem should this species become commercially targeted, and why there needs to be more information about their life history studies. So luckily, there has been some studies about their life history from the Gulf of Alaska, British Columbia, and California. While it seems that it differs, it's important to note that these studies all had different aging methods as well as various maturity indexes throughout their studies. So a little bit about one of the reproductive traits that's found in elasmobranchs. So we have to go back and interpret what happens before multiple paternity. So polyandry is the act of females mating with multiple males. And multiple paternity is where a single litter is sired by different fathers. And this may not happen in most cases. It's found in a variety of taxa from amphibians to mammals, and it's currently found in all viviparous species of elasmobranchs. These are the live-bearing elasmobranchs. So currently, there's only 2 species of oviparous sharks that exhibit this trait, and they are the small spotted cat shark, Silenicus canicula, and the thorny back skate, Raja clavata. But unlike the big skate, they lay singular egg cases. So a little bit more about the big skate reproductive strategies. So it's considered the most fecund elasmobranch known currently, and this is based on earlier sexual maturity of 5 to 8 years, multiple embryos in an egg case, and then being also reproductively active year-round. So it's estimated a single female can lay up to 48,000 embryos in her lifetime. And whether multiple paternity is present due to this trait is unknown. So, as I recalled, big skates can reach up to 2 meters in total length, but these extreme sizes have only been found in their most northern latitudes. So, based on the ecological rule of Bergman's rule, large-bodied animal species tend to live further up north than their small-bodied counterparts, but it's unclear whether reproductive strategies are also affected. So here is pictures of skulls of white-tailed deer in the north, in Michigan, USA, and then in the south, Nicaragua, in Central America. So the objectives of my study was to look at if there's any differences in the reproductive strategies of wild skates in captive settings too. Also looking at if there's size difference in the egg cases, whether Bergman's rule applies to the specimens in the wild, specifically if there's a latitudinal difference in the egg case sizes and embryo numbers per egg case. Also, is there a difference between the sex ratio of the offsprings between captive and wild? I will also include embryonic morphometrics for the developmental stages proposed by a study conducted in 1964, and also create a growth curve for embryos. And then following along, I will use genetic testing to see if there are multiple paternity presence, and whether if one male sires more offspring than the other. So these are my hypotheses. So hypothesis 1 focuses on whether captive egg cases and wild egg cases differ. Hypothesis 2 looks at whether the egg case size differs along latitudes. Hypothesis 3 looks at whether the embryo numbers per egg case differ along latitudes. Hypothesis 4 looks at whether the embryo numbers per egg case are the same in both wild and captive. And then hypothesis 5 looks at whether there is an offspring sex ratio among captive and wild. Then hypothesis 6 looks at whether multiple paternity is present in the species, and then followed by hypothesis 7, where it looks at whether one male sires more offspring than the other. So on to methods. So under the San Jose State IACUC protocol 2014D, I was able to collect 8 cases from the FRAM surveys in 2018 and then from 2014 to 2016. So what I measured was the anterior to the posterior apron of the egg case for their total, for the egg case length, and then width of the main portion for the dimensions of the egg cases. I did not include the horns of the egg cases because those can be easily torn off and then affect my results. After that, I dissected the egg case to determine the fertility status. So if there were embryos present, measurements were recorded such as total length, wingspan, disc width, yolk diameter, yolk weight, and the developmental stage. So the yolk volume was calculated on an assumption that the yolk is shaped like a sphere, where R is the radius or half the diameter, and pi is a constant value. And then the disc width is actually just the wingspan of the animal. So luckily in 1964, Hitz had developed these descriptions to place the developmental stages of the species. So stage 1 includes that there's yolk sac separation, but there's no blood vessel present, and then there's no physical embryo present. Stage 2 is when the embryo is present and there are gill filaments present, but there's no wing development. In stage 2, the wings have developed and there's gill slits present. In stage 4, the wings have developed eye spots and coloration on the dorsal side, and then also sex appendages have formed. So in here you can see the little tiny claspers forming on this male. And then stage 5, the wings have fully developed and the embryo has developed orbital spines for emergence, and at least 90% of the yolk has been absorbed. So I also raised big skate embryos under another IACUC protocol, Jang 1017, and these specimens were acquired from the Monterey Bay Aquarium, and I raised them in the Moss Landing Aquarium Lab from May 2015 to January 2016. I also created viewing windows to determine their developmental stages, and then these species are known to be have an incubation rate of 5 to 6 months. So I collected 25 skin tissue samples from embryos and also from 6 adults, equating to a total of 31 genetic samples. These adult samples were acquired from the Monterey Bay Aquarium. So I also had to use microsatellites to measure relatedness. So microsatellites are simple sequence tandem repeats, and because of that, their high polymorphism is oftenly used in population genetics, and especially used for studies in family relatedness, especially in polyandrous species. So I used 4 microsatellites from a sister species for my study. So here you can see how CTT is repeated 8 times, 7 times, and then 9 times, indicating how polymorphic microsatellites are. So in addition, I had to add a forward primer that includes this M13 tail. This is to allow me to have the fluorescent tags that will be my final PCR product, which I will be which then I can send for sequencing. So then, after all my PCR products were done, I went to the San Francisco State Genomics Transcriptomics Analysis Core, where I could sequence my samples. But before, I needed to use a formalized standard mix, which included 1 microliter of my PCR product, 10 microliters of this high-dye formaldehyde, and 0.5 microliters of this dye standard. So this size standard is used for further analysis for scoring my PCR products. So then these files were converted and analyzed using the GENUS MicroSatellite plugin. Certain programs such as MLRelate, GenePop, Co-Ancestry, and Colony were used for various genetics tests, including population genetics, relatedness, Hardy-Weinberg equilibrium, and parentage analysis. So on to results. So I collected a total of 103 egg cases with 59 fertile and 44 infertile. So here's a scatter plot showing the egg case dimensions with their corresponding embryo numbers. So on the x-axis you have the egg case length, and then y-axis you have the egg case width. And the corresponding colors represent where the egg cases were collected. So here's a box and whisker plot showing you the differences of the egg case length from captive and wild. And you can see that I conducted a t-test where it showed that they are significantly different, with the wild egg case being much larger than the captive ones. And here's just showing you the visual interpretation with the captive ones on the left and the wild ones on the right. So then here's one of our hypotheses where the alternative was accepted, that captive egg cases do not equal wild egg case sizes. So here's more of the samples that I collected, except the egg case length is on the y-axis, and then the locations where all these samples were collected. So starting at 34 degrees north to 48 degrees north, and you can see there's some kind of pattern. I want you guys to focus particularly in around 42 degrees. So what I did was a generative additive model to look at if there was a relationship between egg case length and location. So on the x-axis, you can still see that these are the locations where the samples were collected, and these tick marks just represent where the samples were. And then, but on the y-axis, you have the partial residuals between latitude and egg case length. The gray areas show the 95% confidence interval in this calculation, and you can see at around 42 degrees it condenses. So then back to hypothesis 2, big skate egg cases do change among latitudes. So then let's look at whether embryo numbers occur along a latitudinal gradient. So still on the x-axis you have the locations, and then the y-axis are the partial residuals. Although it was not significant, you can see that there may be some kind of relationship between embryo numbers and egg case length in this location. So back to our third hypothesis, embryo numbers per egg case length don't seem that they differ along latitudes. So here's a bar graph showing you the embryo frequency from the FRAM surveys. So the most common number that I collected was actually 3. So this is 0, 1, 2, and 3. So the most average was 3, and then I did find 8 cases consisting of 6 embryos. So even though I had a smaller sample size, it seemed that 2 embryos from the Monterey Bay Aquarium was the most common in a captive setting. So then back to hypothesis 4, where the embryo numbers per 8 case are not uniform. So here's a box and whisker plot showing you the embryo total length throughout the developmental stages. So I was not able to find stage 1 because most of my 8 cases were far more developed. So you can see how the embryo total length differs among the different developmental stages, as well as the disc width. So to better model this growth, I did a allometric growth curve with the embryo total length on the x-axis and the embryo width on the y-axis. And as you can see, I did a log transform equation with the line fitting most of the curve. But as you can see, there are still outliers above and below the line. So next, the number of embryos that I collected was 219, with 48 females and 37 males. So to determine if offspring sex ratio occurs, chi-squared tests were used with the Yates correction for continuity to see if there was a difference. And the value was, the p-value was 0.54, meaning that it was not significant. I also then tested to see if there were differences among the two environments, and that again, a p-value of 0.66 showed me that there's no significant difference in offspring sex ratios in captive and wild settings. So then back to hypothesis 5, captive offspring sex ratios look similar to wild, to offsprings in the wild. So here's a simplified table of the genetic diversity of the big skate specimens I have, where the first column you have the different loci. So these were the microsatellite primers that I used. And then you have NA, which is the number of different alleles, HO, the observed heterozygosity, HE, the expected heterozygosity, and this is under the Hardy-Weinberg equilibrium. So the Hardy-Weinberg equilibrium has 5 assumptions. 1, there's no natural selection. 2, there's no gene flow. 3, there's no migration. 4, there's no genetic drift. And 5, there is random mating. So it ranges from 0 to 1, 0 meaning no heterozygosity, and 1 meaning high heterozygosity. So a high number represents lots of genetic variability. And usually you want to look at to see if your heterozygosity observed is higher than your heterozygosity expected, because that tells you whether it violates the 5 assumptions found in the Hardy-Weinberg equilibrium. So this table just shows you the different, even with using the same genotype data, genetic programs can differ because of their parameters they use for calculations. So for here, you can see that for half siblings, it differed from 0.2 to 0.4. So this is why I had to take the average in order to make a more better calculation to see what the total relatedness was for my specimens, and it turned out to be 0.73. So these are tables of the paternity results that I was able to match the offspring with the corresponding mother and father. And you can see that both males sired all the offspring, 1 sired 11 and 1 sired 14. So some of these embryos died during development, which made me unable to sex them. And then one of the females laid 9 of the 10 egg cases, she also laid these while she was in quarantine for 3 months. So this suggests that there may be sperm storage in this species. So then back to our hypotheses. Hypothesis 6, multiple paternity is present in the species. So here's just a bar graph showing you the percentages of offspring sired by both of the males. We're particularly looking at the different sexes. You can see that they fall between approximately like 50%. So then hypothesis 7, that one male sires more offspring than another, was not rejected. So here's a checklist of all the hypotheses that we've tested. And then on to discussion. So 8 case dimensions and 8 big skate productivity. So we found out that captive 8 case lengths were significantly smaller than their wild specimens. And this is because the 8 case size is also dependent on the size of the mother. So 2 of the females that were from the Monterey Bay Aquarium had total lengths of 117 to 125. These are actually smaller wingspans found in the wild population, particularly from the Fram surveys, where the females reach at more than 125 centimeters. We also found that embryo numbers in the wild are more than the captive. So why differences in embryo numbers? So one strategy could be to time emergence and limit predation risk. So this is found in green sea turtles, which synchronize their nesting times to increase their survivor rates. So an increased group within a nesting site can create this attack abatement effect, where predators are unlikely to attack a group of prey when the number of prey are greater. So while I did not observe any more than 8 embryos with did not observe more than 8 embryos in an 8 case. One 8 case with 6 developed embryos was found, had an opening. So this suggests that some offsprings emerged earlier before the 8 case was collected. So while the study did not collect more than 18 8 cases in a trawl, large quantities of big skate 8 cases are found, especially up in Kayak Island off the Gulf of Alaska. This amount suggests that big skates may have a different reproductive tactic in their most northern range, which may exhibit the attack abatement effect found in other species. So we noticed that there was quite a few infertility in the 8 cases, which validates that not all 8 cases will be fertilized. So I didn't find any infertile 8 cases among the fertile 8 cases, but when I was raising my big skate 8 cases, there were some infertile 8 cases in the captive ones. So this leads to the question, why would you want to lay a bunch of unfertilized 8 cases, and why invest in laying so many? So laying infertile 8 cases might be an indicator that the females are in a healthy condition. So for, like, let's say chickens do similar things. And then this is a strategy for them to produce infertile dummy 8 cases along the fertile ones to increase predation risks. So predators would choose these infertile eggs rather than ones containing embryos. And these 8 cases are really hard to penetrate, so it will take a long time for predators to actually consume them. So looking at whether there were latitudinal patterns in 8 case and embryo numbers. So going back to Bergman's rule, where we said that species sizes increases among latitudes, with the smaller subspecies in the south and the larger subspecies in the north. So in this study, we didn't see that the species exhibits Bergmann's rule and peaked at 42 degrees north, rather than increasing along the northeast Pacific coast. However, it's important to know that it's unknown whether species up north exhibit this pattern. So, a best explanation for this 42 degrees north is from the California system. So, the California current system is separated into 3 general areas where surface temperature are influenced by seasonal variations. So here we have the Columbia River, which is around 44 degrees north, and then Cape Mendocino, which is around 40 degrees north, and then following by San Diego, which is around 32 to 34 degrees north. So in Cape Mendocino, the winds are the strongest, allowing more sea surface mixing, which increases upwelling intensity. And as you guys know, upwelling intensity increases productivity. So recapping the allometric growth curve, so we noticed that even though the line fit at least 87% of the data, there were still outliers above and below. So these outliers may indicate that maternal investments differ among individuals within 8 casein litters. This is actually found in ovophagus vipertus elasmobranchs, which are found in laminate sharks. So laminate sharks invest in fewer offspring, but additional nutrients for the offspring so that the most fittest embryos survive before birth. The sand tiger shark, however, goes to a more extreme level where they practice adelophagy, where the fittest embryo in each uterus consumes all their siblings until one remains in the uterus. So we did notice that offspring, there were more females than males in both captive and wild. And offspring sex ratio is actually a common evolutionary and life history strategy found in many taxa. One popular hypothesis that proposed this is the Trivers-Willard hypothesis, where it suggests that mothers in good conditions will actually produce more males than female offsprings, because more males would compete with the female offsprings, whereas mothers in poorer conditions will actually produce more female offsprings, because that's the more reproductively stable sex, since the male offsprings will most likely be outcompeted by fitter males. However, this hypothesis is only concerned about ungulates, which have a limited breeding season, can only produce limited amount of offspring, around 2 to 3, and possess only one functioning uterus, which is completely different from the big skate. So is there actually any literature about whether offspring sex ratio is present in elasmobranchs? So in 1982, Charnow looked at this and saw that in spiny dogfish, the offspring sex ratio was about 49%. Ishihara in 2002 found out that one of the females laid egg cases had 56% males and then 43% females. And then in the mottled skate, offspring sex ratios were found to be around 45% in females and 54% in males. So in elasmobranchs, it doesn't seem that offspring sex ratio is known to occur, but many species are known to have sexual aggregations, especially during breeding season. So a little bit about the genetic diversity and population structure. So the genetic diversity of the big skates in my study was quite high, and it falls between the heterozygosity expected values found in other studies, which range from 0.17 to 0.97. The allelic richness was higher than most studies, but this could be because I only used 4 microsatellites compared to other studies that range from 5 to 11. Also, one of the programs, ML-Relate, has limitations due to the minimal number of microsatellites in this analysis, which is why more than a couple programs were used for the validity of these estimations. So I was able to detect one population, which was surprising because 2 of the adults were wild caught. The other adults were raised and transferred from the aquarium of the Bay in San Francisco to the Monterey Bay aquariums. Oh, all right. So genetic relatedness was found in my genetic study. So it was pretty high, with at least 2 of the adults in the wild were related to adults raised in captivity. So high genetic relatedness is not uncommon in elasmoprank population studies. In Larson et al. In 2012, they found out that 65 to 87% of 6-gill sharks were either half-siblings or full siblings in Puget Sound. And this is because Puget Sound is considered a pupping and nursery ground, with siblings remaining in proximity, and this is through tagging and genetic studies. So while we haven't seen if there's any suitable nursery areas for big skates, Hoff in 2010 and 2016 have found multiple nursery sites for 2 species of skates in the eastern Bering Sea. So multiple paternity presence in big skates. As I recall, multiple paternity is present in Viviparous elasmobranchs and other taxa. So therefore, it is not uncommon to have this strategy, but because the big skate is one of only 2 species out of the 300 species of known skates, this is a novel find in the family Rajidae. So we didn't find whether any of the males were dominant in siring more offspring. It was expected at least one male would father more offspring because of their origin. And this is because sometimes lower offspring survivalship comes from captive parents. So this was found where they raised captive animals. Although they may spawn more compared to their wild counterparts, their offspring performed the worst in the wild. And this was found in the hatchery steelhead, Oncorhynchus micus. They were trying to restore native steelhead populations in the Hood River through captive breeding sites. But Christie et al. In 2012 provided evidence that these programs may not be successful for certain species. This actually could be detrimental for zoos that are focusing on, let's say, big cat populations. So for example, O'Regan and Kitchener found that felines in captivity have changes in morphology, and this include bone structure, skull shape, brain size, digestive tract changes, sexual dimorphism, which could be ineffective for these breeding programs. So then on to conclusion. So we found that big skates have a variety of reproductive strategies, particularly in their egg case dimensions, where captive egg cases seem to be smaller than wild egg cases, and the number per embryo per egg case seem to be greater in the wild than in captive. So the average number of captive embryos looks to be around 2, and in the wild looks to be around 3. So we also found out that egg case dimensions and embryo numbers peaked at 42 degrees north. And then we found out that multiple paternity is present and sperm storage is present in the species. So for future studies, what I would really like is to collect specimens in the most northern range to determine if Bergmann's rule is applicable to this species. And then second, although my genetic studies revealed a high heterozygosity in the species, I only had a limited amount of samples. I would really like to have a more detailed survey about big skate populations in the Northeast Pacific, particularly focusing on their genetic diversity range and where the populations are structured. This would provide information should any aquariums want to implement future breeding programs. And with that, acknowledgements. So I really want to thank my committee members, especially Dave, for actually letting me into this lab at first. Thank you for all your support and just keeping me on my toes to make sure I finish. John, thank you so much for all the genetic help and letting me use your equipment. And then Scott, thank you so much for giving me like a more broad approach of how to, what would you say, look at the bigger picture, because I was so focused on what my data represented, and you were like, oh, hey, let's look at all these different studies. And that really helped explaining some of the patterns I saw in my thesis. I want to thank a lot of people for the specimen collections, particularly people from the FRAM surveys, and also people from the Monterey Bay Aquarium, including Manny Ezekura, Michael Howard, Kelsey Barker, also Larry Young from San Jose State for getting my IACUC approved. And then for genetics help, thank you Sean Larson from the Seattle Aquarium for all the help, and Jason Smith, April Woods, and Holly Bauer from Norte for trusting me with their equipment late at night. I also would like to thank Colin Leisure from San Francisco State University for the genotyping services, as well as my UROC interns, Charlie Farber, Franco Sanchez, and then the Marine Pollution Lab. Thanks Autumn and Wes for being so flexible with my work schedule so I can finish my thesis. And then for all you first years, one thing you guys should know before is to learn how to program, not when you get your data. It is so painful. So these are just a limited number of all the people here available that know their programming. Get to know them. And I would also like to thank my lab mates, Melissa Niemans. By the way, she's presenting her thesis at 4 o'clock. Don't miss it. Vicky Vasquez, Justin Cordova, Marty Schmidt, Amber Reichert, Sophie Bernstein. My funding sources, UROC, the David and Lucille Packard Foundation, the Myers Trust, and the WAVE Scholarship. And then last but not least, my family, even though they're not here, so I get to poke fun at them. So especially my mom, because every time she calls me, she's like, are you done yet? So now I can finally say, I am done, and I'll take any questions.
Jessica?
That was really great, really interesting, Jess. I was wondering for the samples, the skate cases collected from the aquarium, I know you said it came from the Aquarium Pacific, but where did they get those before?
So I think some of the specimens, I believe, they collected from the wild, which they raised, and then they started their own breeding program. And then I guess due to this whole trying to start, a breeding program for big skates, it's proven that it's better to have captive raised individuals because of all the permits they have to get.
Yeah, so I'm wondering if you think, because if there were latitudinal changes for the, wait, did you say you did find latitudinal changes?
It was
for the
at
wild
42
ones?
degrees, but my samples were only from Washington down to San Diego, because originally I was trying to collect some from Gulf of Alaska to down to British Columbia, but we faced a few obstacles, permitting issues. It's always fun, right? Yeah.
So, is it correct that you genotyped 25 embryos and 6 adults, and we believe that all of the embryos are offspring of those 6 adults?
Of one of the females and of 2 of the females and 2 of the adults. What I didn't include was at Colony, the paternity test program had suggested another male involved in the siring, but during the whole period where the female was seen copulating with the other males on the exhibit, there was no way to have that pseudo male.
So there's 4 mothers and
There's 2 mothers.
2 mothers?
And then 2 fathers.
Okay, so if they were all heterozygotes, then there would be 8 alleles possible in the remaining adults. If they were heterozygotes and they're all different genotypes.
But I think 2 of the adults were related to each other. I think they were half.
Okay, but maximally.
Yeah.
Maximally, there could have been 8 alleles in the leading group.
Yeah.
Yet you saw 15.
Yeah, so
alleles.
that's why I think it's because due to my limited amount of microsatellites, if I had a couple more, I think that would.
But that's on a per locus basis. Per locus, you saw around 15 alleles per each locus. So just taking each locus one at a time, that you would have expected a maximum of 8.
So then that could say that something's wrong with the.
So I'm suggesting that there may be an issue with the scoring
Or
of the
the.
microsatellites. So I think you need to review the chromatographs, maybe by eye, and make sure that the scoring in the software system makes sense.
Okay. All right. Thank you.
Any other questions for Jessica?
This is just a big simple thing, but when you showed the 2 egg cases and one was blue, was that like a computer scan of an egg case?
Was at the beginning.
Oh. For the different that species, because I was comparing the different species.
Yeah.
Oh, genus, yeah. Yeah, that species during Polka has pretty weird egg case colors, right?
Yeah.
Oh, so that was a natural color?
Yeah.
Any questions?
Is there any evidence of cannibalism within the egg cases?
I don't think so. that I raised.
Oh, I have a question. Would it be possible to, you said that you couldn't determine the sex of the other egg cases. Isn't there a way to genetically determine sex in those? Or?
In mammals, you have this, John can help me with this, the Surrey gene, right?
Okay.
That's used to detect whether it's female or male, but I don't think we found one for elasmal brains.
Okay.
The Surrey, so for detecting sexes. Yeah.